All right, everybody, so we're going to go ahead and get started, and I'm sure other people will filter back into the room. So we have the post-lunch crowd. Hopefully everybody will be able to stay awake after lunch. So I'm here today to tell you a little bit about TreeScan analyses. So our morning really featured inferential designs, and they were designs when there was one exposure and one outcome of interest, and you were really seeking to find a risk estimate associated with that particular exposure outcome pair. So TreeScan takes us in a bit of a different direction. So TreeScan, we're going to start with sort of the basics of it. It is a signal detection or data mining method. And so here, the difference between what we talked about this morning, where you were focusing on one exposure outcome pair, is now we're going to open it up and we're going to say we're not going to limit to one outcome. We're going to look at a set of many, many outcomes. And in this case, more than 8,000 outcomes. And so what this type of an analysis is meant for is screening, similar to other sorts of hypothesis generating type mechanisms. So some key features about TreeScan, in addition to it being hypothesis generating, it also adjusts for multiple hypothesis testing, because in fact, you're testing as many hypotheses as outcomes that you have available to you, and in fact, even more than that, and I'll explain later why. Additionally, what TreeScan does is it takes electronic health data in claims, and it not only looks at individual outcomes, but it actually leverages groups of like outcomes. And the reason for that is then you don't need to actually understand how a clinician might code something, but you have a group that's clinically related, and you look at all of the counts for that entire group when you're actually doing an assessment. And so I want to bring this back a little bit also to some of the learnings that we had this morning, because where TreeScan is different is that it adds multiple exposure outcome pairs, but it actually leverages the same designs we already spoke about this morning. So TreeScan actually comes in two flavors, the first of which is a new user cohort design with propensity score 1-to-1 matching. So just like you learned this morning, you have Group A, or Drug A, and Group B, Drug B. But then instead of looking for one particular outcome, we've expanded it now to many, many, many outcomes. So it takes the same epidemiologic design and brings it into many more outcomes. Similarly, there's a self-controlled risk interval design, and it's the same concept. You take that one exposure, but now instead of looking for a single outcome, you look for many, many, many outcomes. And so the reason we chose to place the TreeScan training here after you've already learned about the designs this morning is because it leverages both of those designs and expands it to do many outcomes. But one thing that is the trade-off in expanding to many outcomes is then you cannot control for one particular outcome. So there are different considerations in your specifications and your trade-offs, because when you're only looking for one exposure outcome pair, you take a lot of care to deal with confounding related to that outcome. And because confounding is by definition related to a particular outcome, you can do a lot more. When you've opened it up to now look at upwards of 8,000 outcomes, you've lost the ability to deal with that specific element of it. So there's a use of the known epidemiological designs, but there's also the consideration that when you relax some of the parameters in order to allow you to look at these outcomes, that you can't do as fine of confounding control as you could if you had just the one exposure outcome pair. And so there are several different types of designs that we use with TreeScan. The one I'm going to talk about today sets one exposure and then looks at many outcomes. And the way that we do this is using something called the multi-level clinical classification system, which was put forward by the Agency for Healthcare Research and Quality. And what that does is it groups like things by body system, where there are many, many outcomes to look at. And I'll show you an example of this tree. So here, let's just talk about some concepts behind what is a tree. How is data structured like a tree? We start out at the highest level, and we call that the root. And then as we get to finer and finer and finer bits of analysis, we get down to the leaf. So essentially, the most general is at the root, and the most specific is at the leaf. And so for folks in the room who may be familiar with clinical trials or also with the FAIRS system, the Adverse Event Report System, the measured reporting terms are also in a hierarchical tree. So the root equivalent here is the system organ class. And then as you move down the tree, you get to the more fine-grained information. And in fact, many analyses performed, at least using the FAIR system, are done at the preferred term level, which in our tree here is the 4th level if you're counting from the top from 1 to 4. So just how MEDRA terms are structured into a tree, the multi-level clinical classification system is also structured into a tree. And then there is even a third therapeutic classification system that I won't talk about today, but it structures drugs also in a tree. When you can imagine that there are classes, and then there are particular entities, and then within those entities there are particular dosages and strengths. So this is a very simplified example of just part of a tree. And so in an exposure-oriented scan, you set the exposure of interest, and then this is looking at how these things are grouped together. So you see at the top is a very general category, diseases of the nerve and sense organs. And then when you go down to the very last level, which we would call the 5th level or the leaf level at the bottom, that's going to have the specific ICD-9 and 10 outcome codes that we're searching for in the data. And you can see that on the bottom there are 5 outcome codes. They all relate to convulsions, but they're all slightly different versions of convulsions. And so the idea here behind looking at a tree is that you might know that you're looking for convulsions, but you might not know exactly how physicians code them. And of the 5 available coding on the bottom, it might be that one is used preferentially in one health system and one is used preferentially in another. The key about TreeScan is you don't have to know that ahead of time. You don't have to know that a priori, because you're going to look at all of them and all the combinations of them. And it's in doing that that you actually can potentially find where the particular risk is. So I said at the beginning that TreeScan is a signal detection or a data mining method. So it's important to understand sort of equivalent signal detection and data mining methods used by the FDA. And so the FDA uses spontaneous reports in the FAIR system. And so I want to talk a little bit about how TreeScan is similar and also how it's different than FAIR's. So first let's start out with how it's similar. So both of them intend to be a general safety net, meaning that again, there's no need to specify a particular exposure with a particular outcome pair. The idea here is you're searching across all of these outcomes, looking for any pattern or frequency of unusual behavior. Both of these techniques are intended to generate hypotheses. They are known as not being hypothesis-driven. So they produce hypotheses as an end result of the analysis. And they test many hypotheses to get there. Both, again, use the data that is structured into trees, just as I just talked about, with the MEDRA terms being structured in a tree and also the clinical outcomes being structured in a tree. But then how are these systems different? The most prominent difference is that they utilize very different data sources. So the FAIR system utilizes spontaneous reports. And for folks who are familiar with spontaneous reports, those occur when either a patient or a provider or industry submits a report to the Food and Drug Administration where they believe that an exposure may have caused an outcome. And so there is a need to link those two in your mind in order to actually have the report. That's contrasted to observational data, where we're going to look at all new users of a particular exposure, regardless of whether we think it may or may not be related to an outcome. And that's often referred to as longitudinal data or having denominator data, meaning you have complete capture of an entire population, which allows you to get to rates. The other key feature here is it again allows you to use the epidemiological designs we learned about this morning, the propensity score matched analysis that we talked about early this morning, the self-controlled risk interval design. Both of those depend on having complete capture of an enrolled population so that you can account for every single person that may have been exposed to your exposure of interest. Finally, these produce very different analytic data sets. So the results of a FAIR's analysis are that you get a case report, and that case report often has very detailed information about a particular patient and all of the things that are happening to that patient. Here in TreeScan, you're really just getting counts. It's very aggregated, and it's done so on purpose to be privacy preserving and to allow it to operate effectively in a distributed network. So back to this, this diagram to specify that we, in fact, use two different designs with TreeScan. We use the one-to-one propensity score match design, and we also use the self-controlled risk interval design. And I'm going to talk to you a little bit today about the self-controlled risk interval design. So that's going to be what we discuss in terms of specifications. And you'll see a lot of overlap with everything that you just learned about this morning with Justin. So let's recap the self-controlled risk interval design, the main advantages, as Justin highlighted this morning. Self-controlled designs control for time invariant confounding. So when things about you don't change over time. and they're very easy to implement. But they are very vulnerable to time varying confounding. And the other issue is because you're comparing periods. If you have somebody who has a sustained elevation of an outcome over the entire period, you actually won't pick it up, because the whole point of the self-controlled risk interval design is to compare period to period. So if in fact you have an exposure and an outcome where the outcome is elevated for the entire period of both the risk and control window, you won't detect it, because what you're looking at is a comparison among periods, not for that sustained risk. In those kinds of situations, the propensity score match design or the new user cohort design would be a better fit. Another thing that I wanted to talk about some of the conditions that Justin went over this morning about what are good examples of when to use a self-controlled risk interval design. And particularly he talked about having a fixed event. So in this case, we're going to go forward and we're going to talk about a fixed event. And that fixed event for us is going to be a new start of statins. Now, statins, as you know, are a prescription that people will hopefully continue to refill and use again and again as a chronic med. Here, we relax the criteria to some extent of having to have a very fixed event. And the reason is because we're doing a screening analysis, and also because we're keeping it very short. So our outcome window that we're looking in is a very short period of time where we presume that a new user would still have influence of that first start, whenever that would be. But an important point to point out here is that we aren't actually accounting for the duration of therapy in the designs you'll see today. And we do that on purpose because of trying to keep it simple, and because we are moving away from a particular design for exposure outcome and instead looking at all outcomes. So we're going to monitor single ingredient statins for 8,000 plus outcomes using a self-controlled risk interval design in our training example today. So first we're going to go through our stoplight rubric as we've been going through. So single ingredient statins, these are self-administered. People pick them up at the pharmacy. And as a result, those are green in our system. So we're going to have easy ability to get at those dispensings. So which ones did we monitor? We looked at 7 different single ingredient statins. They're listed there for you. I also have the ones that are noted that are deemed in the high intensity category. That will become important later, but for right now, I'm just noting them there for you. So why did we choose to look at statins in this training example? One, they have a very large sample size, and they have an established safety record. And so we can look at something like 8,000 outcomes and expect to see something that corresponds with what we know about those particular drugs in real life. And so we chose these in particular also to look at the idea of looking at a fixed point, and in this case the fixed point is the first start of the statin, the new use of the statin. So this is how we build the design, and this is going to be very much review from what you went over this morning, so I think I'll try and go through it a little bit faster. So the first thing we do is, again, we look for, and let me stop first here and say, as I go through the building of the cohort, so oftentimes when we do trainings or we do even scientific lectures, we have a methods section and then we have a results section. So you present all the methods and then you present all the results. Here I'm going to kind of blend them both together a little bit because we're going to build our cohort and then I'm going to show you a cohort attrition table right afterwards. And the reason is because I want you to understand in the observational data where we're actually losing people and why. And so I'm going to continue to flash the building of our cohort, and that's effect in the attrition table, and go back and forth and back and forth. I just want to prepare you that it's probably a little different than normal scientific presentations where you see the attrition table put all the way in the results section. So with that being said, let's start out here. We're going to start for looking for our single ingredient statin of interest, and we're going to check to see that we have new use of that statin, so that nobody has had a prior dispensing of that statin in 183 days prior to the exposure. So here I'm going to tell you about the largest group of statins that we have, which is simvastatin. We did this training in 35% of our Sentinel distributed database. Because statins are so widely used, we actually didn't need to use all of the database. So what you see here is actually a subset. So the number of total statin dispensings, regardless of new statin dispensings, was over 60 million in the query period. And then when we implement our new user criteria, we lose about 90% of those dispensings. And the reason is because people should be taking statins continuously. And so you see this large dropout, it's as expected. It's actually as desired, because you want to see people who start on this drug continue to take it. But what that means is that you end up with only 6 million new incident users at that time. Once we figure out that we have incident users, we're going to look for our enrollment criteria. And so the enrollment criteria here is going to have a pre-exposure enrollment, and it's going to have a post-exposure enrollment. And additionally, you have to also be enrolled on your index day. And so the reason for that in the pre-exposure period is to ensure that you're looking back for the same period of time you looked for your washout period, so you have complete coverage of those individuals. And the reason for the post-exposure is because you want to have somebody to be eligible to have the outcome on every day in that observation period in order for the null hypothesis and the biostatistical pieces of it to work out. And so here I want to show you that when we impose that enrollment requirement of 240 days of enrollment with a very particular sort of breakdown of which has to be pre and post index, we lose a good portion of the cohort. One of the things that's true about statins is they're often used by individuals who are transitioning to Medicare. And in our data, when we look at the individuals that we have, what is true is that we don't have an ability in database 1 to know that the same individual has data in database 2, because we don't force database 1 and database 2 to talk to each other and share information about that individual. So we do have some drop-off when individuals age into Medicare, in that we see their data in database 1, but then we don't know that they're in fact the same individual in database 2. And so it's important to understand that when you're looking at individuals that are in this particular situation, that you may have more loss to follow-up than you might otherwise expect. Additionally, so when we lose that, we end up, we're down now to 1.7 million individuals who have a new incident dispensing of simvastatin and have the required enrollment coverage for them to continue in the study. Now we're going to actually look at excluding individuals for particular reasons. And so in our study, we required people to be adults, so they had to be 18 years or older on the index date. I guess that should say or older, or have a plus by it. We also were looking at trying to eliminate time-varying confounding. And so we didn't want a recent history of PCI or CABG or coronary thrombolysis. And the reason for this is because statins are administered for two reasons. One is primary prevention, and that was our target population. We wanted to find individuals who go to the doctor, who have an initial indication that they have high cholesterol, and then get dispensed a statin. But statins are also given as secondary prevention, meaning that if somebody has a cardiovascular event for which they have to be hospitalized, they are discharged with a statin. And so what happens in a self-controlled design is you see the effects of their prior hospitalization carry into the observation period after the exposure. It's not related to the fact that they got the statin. It's related to the fact that they had this hospitalization and these cardiovascular procedures prior to the statin dispensing. And what we're trying to do with these exclusions is eliminate that population so that we don't have that time-varying confounding in our study design. So when we do that, we lose a very small proportion of additional people, and we end up with our exposed cohort of around 1.7 million individuals who contribute, who are following at this point. Then we're going to detour a little bit from the design that we talked about earlier with Justin, because in TreeScan, actually what you do is you look for an observation window, which is an amalgamation of both your risk and control. So you just put it all together, and you just specify the start point and the end point. And in that window, you're going to pull out any outcome that you define in whatever setting you want. So we define the emergency department or the inpatient setting as a proxy for something serious, but you can actually use any setting you want, and you can make it more restrictive if you want. And we're going to look for all potential outcomes in that window, and we're going to record the dates of those outcomes. Then what we're going to do with those outcomes is we're going to check that they're the first outcome in a particular period of time. And so this is very similar to what Justin showed before. So if you see an outcome, you want to know it's the first outcome in a certain amount of time, because you don't want to be following somebody for some pre-existing condition where they continue to have that outcome and you're just seeing the natural progression of that history. And here, this is where the tree comes into play, because not only are you going to check that the outcome is incident with respect to itself, but also any clinically related grouping of outcomes. So how does that work? The clinically related grouping, we're going to go back to our demonstration before. If I see an outcome, for example, on my screen of convulsions, which is all the way to the left and is one of the ICD-9 codes. If I see that outcome, and then 2 days later I'm hospitalized again, and there in the hospitalization I get the other convulsions outcome all the way on the right side. So now I have two outcomes in the same clinically related grouping within several days of each other. It's likely that those are related to the same underlying condition and that they're not two isolated events. And that's the reason why checking that the outcomes are incident to anything clinically related prevents you from false positives, from basically following somebody who is contributing multiple times for what is really underlying the same condition. And so this is an example of a clinically related grouping, and we do many clinically related groupings in TreeScan to ensure that we're looking at the right unit of analysis. And so when we do that, we end up with 405,468 outcomes that are eligible to be included in our analytic data set. So just to recap, we have 1.7 million users of statins. These are incident users of simvastatin in this case, who have the required enrollment period. And when we follow them for the 56 days after their new statin use, we pick up 405,468 new outcomes that they have not had in the prior 183 days, and outcomes where they haven't had a similar or related outcome in the prior 183 days. And of those outcomes, they are spread across the tree. And the tree itself is more than 8,000 outcomes large, if you would. And so what this really is a screening analysis, a complete analysis looking for anything that is eligible to be included in the analytic data set. And so on my slide, you haven't selected the risk window. So Justin talked to you before about selecting the risk window and sort of the importance of doing that in the self-controlled risk interval design. Well, when you have only a single outcome, then you might have some clinical information to suggest something about biological plausibility or about when that outcome could be expected to be seen. But when you have 8,000 outcomes, you have to select a single window that applies to everything. And that's basically impossible with a risk window. And so what we do instead is we select an overall observation window. And then what we do is we scan that window. And so there's a little animation here to show. We let that risk window move across the observation window, and we check every combination that is legitimate based on some user-specified parameters. And what we're looking for there in that scan is where is the peak activity of interest. So where is the peak amount of occurrence over that entire period? It could be in the first 7 days after exposure. It could be in the last 7 days of the observation window. You don't know beforehand, and you actually don't specify in this analysis. You allow the data to be tested, and then you take the outcome that is most frequently occurring using a way to control for multiple hypothesis testing to find when it's most of an issue. And so there are thousands of hypothesis tests, because if you start out with 80,000 outcomes, and then you add groupings of related outcomes, and then you add different risk windows, you end up multiplying to the point where you have more than 1 million hypothesis tests. But, good news, they are highly overlapping, meaning that you don't have to do something more complicated for false positive control. And you do it using a simulation setting so that you completely control for all of that multiple hypothesis testing. So this is the part where we get a little statistical here. The null hypothesis here, when we look at this, is that there is no outcome or grouping of outcomes that are occurring during a particular risk window within the observation window. What that means is we expect these outcomes, if they're completely unrelated to the exposure of interest, to occur at any time. There should be no reason why they're occurring preferentially at any point in time following the exposure. And so if we see evidence to the otherwise, then we reject the null hypothesis, and we call that an alert. So what is a TreeScan alert? An alert is a statistically significant finding of greater than expected occurrence of an exposure outcome pair while controlling for all that multiple hypothesis testing. It's really important to note here that an alert is not a signal. And the reason is that there are a lot of things that are explainable temporally based on the exposure of interest and the outcome. There are known and expected things. I'll get to that actually in the next slide, so I'm previewing my next slide too much. The point is that when we get alerts, we have a way to triage that information. And there are only some alerts that we deem to be the later signals. And in fact, it's FDA that creates that classification. And once we do that, we go ahead and follow those in other pharmacoepidemiologic methods. So how are alerts triaged? The first very easy pass is the outcome in the label? Does it already say in the label that we expect this outcome to occur? And so we've done this before with various vaccinations, and almost all vaccinations have an outcome of cellulitis or rash at the vaccination site in their label. And so when we see rash or cellulitis in the data, it actually is a little bit comforting because it tells us that our analysis is working the way we think it should, the way we intended. We also want to rule out possible adverse reactions due to time-varying confounding. So this relates to an example that also Justin featured before, which is when you have a PPSV vaccine, it's given prophylactically prior to spleen surgery. So could you say that the vaccine, in fact, causes the surgery? Well, temporally they're related, and it makes sense to see them in time. But you're not suggesting that, in fact, there's something about the vaccine that biologically causes the surgery. And then once we've done that, we narrow down to a list of possible adverse reactions that might merit further follow-up, which is when we consider addressing them in alternative study mechanisms or tools. And at this point, now you've narrowed down to a particular exposure outcome pair. So now you can bring into play all the information you know that might be outcome specific. Then your confounding control then can be outcome specific. So this is a design diagram. This is not drawn to scale. It's just drawn to show you that these are all the parameters, in fact, that you need to specify for TreeScan. Everything that you see here that's underlined is a parameter, meaning that in the program itself, you enter in the value that you want it to have and the program will operate. And anything in brackets is also a parameter. So the only remaining parameters are then the code lists that describe your exposures and your outcomes. So in the end, there's actually not that much to specify to do a very good screening analysis. So now we'll look at results, since I already showed you the whole attrition table. Here is the attrition table we just saw. Now it's in entirety instead of as part of a build. Here's the data. So we said we found 405,468 incident outcomes following new simvastatin use. This is a histogram that we just throw up there. I always do this to look at the data because I think it's important to look for the temporal pattern ahead of time before you know any of the particular outcomes. You'll see here that there's still a lot of outcomes that are occurring immediately following statin exposure, days, for example, 1 to 9. And that's important because what we were trying to do by including exclusions for recent hospitalizations related to cardiovascular outcomes was we were going to try to eliminate looking at outcomes that are directly related to that hospitalization, where the statin dispensing itself is incidental. What this tells me when I look at that is we didn't get all the all the procedures we were looking for. There are other procedures going on that are continuing, people are continuing to be followed in that time immediately after their statin dispensing that we didn't actually include in the exclusions. So it's important to look at your data for those reasons. So this is a big table, so we're going to take it in chunks. First, the overall is that the simvastatin produced 11 alerts. So these met our criteria in order to be considered worthy for further triage. So I'm going to start with the top and I'm going to read across this information. So we start with the node name. So remember, this is a tree of 8,000 outcomes. So the node name is the name in English of the outcome. The node ID is actually the place on the tree where it exists. The node outcomes, and I'll walk through with this, so let me just say this. So the top hit, if you would, from our TreeScan is that folks with a new dispensing of statins have a higher frequency of unstable angina. That higher frequency occurs within a TreeScan detected risk window, and RW here stands for risk window, of days 1 through 7 following dispensing. And in days 1 through 7, there are 523 of those outcomes. And that is relative to days 1 through 56, where there are 2,269 outcomes. So what it's comparing is, again, remember, the idea here is that you're comparing time periods. And if there is no exposure-outcome relationship at all, then essentially an outcome is equally likely to occur on any of those 56 days. So if you see a greater occurrence in a particular interval, then what it does is it tells you something's going on here. And so here we see this greater occurrence in days 1 through 7, and then we have the associated test statistic and p-value. And so we go through and we look at the alerts for each of these categories. So this is one alert. I want to bring your attention to another one, which clearly is showing that time-varying confounding I was talking about. So here the alert is hemorrhage or hematoma complicating a procedure. So here, what it's telling you is there had to be a procedure. It's one that we're missing or one that we're not getting in our data. We don't see it. It happens prior to day 0. They have a procedure. They're sent home and dispensed a statin, an initial dispensing of that statin the first in 183 days. And then after that, they have a hematoma. The hematoma is not necessarily related to the statin. It's related to the procedure that actually caused somebody to dispense on a statin. And it's here that we see that when I was saying there are things we should have excluded that we didn't know to exclude, clearly. This is where this is coming out. And this is why we call these alerts, because it's screening, and we know that there's a possibility that we miss something. And so we need to go through each of these alerts and consider them. But again, not every alert will go on to become a signal. There's a lot of clinical judgment involved in looking at these data and understanding whether there's actually something there that is worthy of potential follow-up later. So I'm just going to show you another cohort that's very similar, only to show you that it's really very similar. So this is the second of all the single ingredient statins we have. Simvastatin has the largest capture. The second largest capture is atorvastatin, which is shown here. You'll see if you look through sort of the numbers that everything falls about the same, meaning you lose about the same amount of people because they're not incident dispensings. You lose about the same amount of people percentage-wise because you don't have the required enrollment. The only thing that's really different is that you lose more people relative-wise in the exclusions. Here you lose about 3%. In the simvastatin, it was closer to 1%. And the difference is because atorvastatin is one of those high-intensity statins that I showed you at the very beginning. And so for people who are having a procedure, a cardiovascular procedure, other procedure in the hospital, and who are dispensed a statin, the actual clinical guidance says that you should preferentially dispense a high-intensity statin. And so you see more of these folks excluded here because preferentially the clinical guidelines tell you that you should be dispensing a high-intensity statin versus a not high-intensity statin. So here's our atorvastatin pattern. Here you can see that we still have the same sort of stickly issue with those days 1 through 9 showing up stuff that we would otherwise not think should be there. I should note that the increase in days 1 through 9 could be because there's actually really an elevation, but because this is capturing every outcome, it's unlikely that one outcome would drive the entire picture to be higher there. So it's important to keep in mind when you look at this diagram, you're looking at all the outcomes, everything that registered, not necessarily anything that's particularly notable. And so here with the atorvastatin, this has 8 alerts. And again, simvastatin had 11. But it's important to know that all the alerts here are entirely represented in the simvastatin alerts. So everything that was there is repeated here. And you've lost a few of them. And the reason you've lost them is actually because you have a smaller sample size. So what it tells us, though, is very internally consistent, is that people who use single ingredient statins have the same patterns of care. They have the same outcomes that follow those initial dispensings of statins. And so you see that top hit is identical to what it was before. Here you have a longer risk window. So here it identified days 1 through 15 instead of days 1 through 7 because the period of more elevated activity is different in this case. And you can see that throughout. It depends, again, on the mix of the sample size and the fact that the software itself is built to detect the highest probability of an unusual occurrence. And again, here we see our hematoma still coming back. So some other statin results. So I said we looked at 7 single ingredient statins, and I showed you particulars about 2 of them. This is the data on the other 5 single ingredient statins. They're ordered from the largest cohort to the smallest cohort of the remaining 5. The exposure cohort is the number of individuals that qualified to actually have any diagnoses in the 56 days following their initial incident dispensing. The analytic data set is the number of outcomes that actually met the criteria to allow them to be measured. And the alerts there shows the number of alerts that followed when you do the same result here. And so in the largest cohort, you only pick up that top number 1 alert, unstable angina. And after that, you pick up none of the alerts going down, and that's really related to power. You don't have the power to find anything that's more than that. We did a sensitivity analysis to look at a different level of aggregation. So what I mean by that is going back to the tree, and actually I don't think I have a picture of the tree, so I'm going to go all the way back to the tree. Sorry. This here is the default level. The 4th level is 1, 2, 3, 4, counting from the top. The 3rd level is if I raised it up and I said, well, now I'm going to look at a bigger clinical grouping. Here, epilepsy and convulsions clinically are quite different. So it's maybe not advisable to go up a level, but in certain other parts of the tree, there's not as stark of a difference. But the point is it's user specified, and that you can do it in a sensitivity analysis to try to understand whether there's a bigger signal happening potentially in a bigger grouping. So I'm going to speed through this. We're going to go back to this. So folks may be familiar that there is a known adverse reaction of statin-induced rhabdomyolysis, which I'm not a clinician. I know there are some clinicians in the room. I'm going to say is muscle weakening that becomes more severe. And so there's a study that was performed several years ago by folks here at FDA, and they found a mean time to onset of rhabdomyolysis of 348 days. And so just as a visual to show you, that 348 days exceeds our study period. So if you set your observation window from days 1 through 56, that's what you're going to find. You're going to find outcomes that actually have onset in days 1 through 56. So you have to think about when you're setting your observation window, taking those kinds of things into account, the limitations of your study, that you're not going to be able to see something that has a long duration time of onset and then shows up on day 348. So some statin takeaways. No new or unexpected alerts were identified. All the alerts that were in that list are all clinically explainable given the drug's use as a secondary prevention following hospitalization. It suggests that the method itself is able to filter out noise. So again, of the hundreds of thousands of outcomes that were picked up, you still only ended up with 11 alerts at the end of the day to actually go through and see, do we think that this is something related to the statin or not, which is pretty manageable. But despite exclusions, there was still some lingering pre-existing disease, and that is best seen in the fact that one of the alerts was the hematoma following a procedure. And so what we would strive to do is actually exclude as many of those recent hospitalized procedures as possible so that we would eliminate those kinds of things. Because really our target population here is to understand more about potential adverse events following the primary prevention utilization of statins. The other problem is that when you have that large number of outcomes that occur as a result of some prior condition, it can drown out or mask something. So you can think of it like an iceberg. And so essentially what you want to be able to do is reveal as much of the iceberg above the surface as possible. But if you end up with a bunch of information in there that's really not helping you, it can push that iceberg down and then you can miss something that's occurring underneath. So some limitations here. And these are very similar to the limitations Justin went over earlier. With a self-controlled design, you depend on onset times that are available in the data model. You cannot distinguish a sustained elevated risk of an outcome. If you want to do that, actually the new user cohort design with a propensity score match 1-to-1 is a better option for you. And that you're vulnerable to time varying confounding. Some of the pieces of this, though, that despite these limitations still make it useful is that it's very easy to implement. There's very few parameters and it can be done very quickly, and you get data then on 8,000 plus outcomes. So with our current TreeScan software limitations, we look for acute outcome events only with a fixed follow-up, so in this case 56 days. We have a new version that we're in the middle of QCing right now and putting into production. It's going to allow varying follow-up in the post-exposure period, and that's going to be available in 2018. So what that's going to do is it's going to lift that post-exposure enrollment requirement and allow any degree of post-exposure follow-up time that anybody has. It will remove a potential selection bias because now people don't have to be enrolled for the period following their exposure, and it will increase the size of the cohort and the time available for study. So in summary, TreeScan supports multiple common study designs, but you have to choose the right design for the right product. And there may be times when the one-to-one propensity score match design is superior to the self-controlled risk interval design. In the same way this morning when Justin and Jane both talked about it, there are times when you would want, even in a one exposure, one outcome type look, to choose one of the designs over the other. The self-controlled TreeScan methods perform best when they are applied to stable patients. So for example, those who are starting a contraceptive or a vaccine, those who are starting statins for primary prevention. It has moderate performance, or you get more unwanted time-varying confounding when you have patients that are not as stable. So maybe those who have been recently hospitalized or are being recently followed for some new health problem. And it actually performs pretty poorly when you have acutely ill or unstable patients, and it's precisely because of that vulnerability to time-varying confounding. Because what you're going to see in the period that you're observing are all of the things related to their acutely ill status, as opposed to potentially the drug that they've just newly been dispensed. And so I want to point you to the website where we have the software available for you. So here, this is a screenshot of the Sentinel website. Up at the top, you see that there's going to be a section on data and surveillance tools, and then within the data and surveillance tools, there's a place called software toolkits. You'll see that it comes just below the modular programs, the routine tools that we talked about this morning. And then if you click in that software toolkits, in there is tree extraction documentation and SAS programs. So I should specify here that this is actually a two-step process. Tree extraction is what we use when we want to gather the data that forms the analytic data set. And so this is the data. And so basically what this program will do is if you have data formatted in the Sentinel Common Data Model, you can execute this program against that data, and it will draw for you that analytic data set where it will look for new users of a particular medicine or procedure, and it will look for your enrollment requirements as discussed, and then it will pull out the outcomes, the analytic data set that you use for tree extraction. Once you get that analytic data set out, then you actually go to TreeScan, which is a publicly available software. And so I did a talk at ICPE this last year, for those who are familiar with it, and I taught a little workshop on how to use TreeScan, which is actually the freely available software. So again, this is a second part of a two-step process. I have some fake data sets here. They're all simulated data for folks to practice on their own. In addition, if you click on the materials here, you can get to the presentations that were there that explain how to actually use TreeScan itself to perform the analysis on your own. So again, two parts here. The tree extraction piece, that requires your data to be formatted in the Common Data Model and to utilize the SAS program to get at that data. And then TreeScan, which actually performs the analysis. TreeScan itself is actually agnostic as to how you get the data. So if you get the data on your own, using your own program, you can still run it through TreeScan. It doesn't matter that it has to be Sentinel formatted or anything like that. And TreeScan is downloadable to all the different platforms. And with that, I might be a little early, and I will take any questions.
Hi, Charlie Barr. So you'd mentioned that sample size has an effect. And so two circumstances in which I think that might be applicable, and I'm interested in your experience and insights on the issues and recommendations. So the two situations would be if you're looking in subgroups, population, so it's going to be smaller sample size, or you're trying to look at individual products or manufacturers to see if there's a different rate.
Yeah, so sample size is always something to consider. A feature that is in the analysis phase, not in the design phase, in TreeScan actually has a power evaluation feature in it. So you can understand how much power you have for a given frequency of an outcome. So the way that that looks is that you would look at your data and you would say, you know, if the outcome is occurring at 1 per 10,000 person years, what's the probability that I would see that outcome if it was elevated? And so the power evaluation allows you to do that if you have some set rates at which you're sort of benchmarking what you can do. And so there are a couple of potential solutions. My colleague Martin Kulldorff, who's actually the author of TreeScan, is actually working on a means to do it sequentially, where you control for multiple hypothesis testing not only over, again, the tree and all of the intervals, but also over time. So you could run TreeScan, for example, at time point 1, and then 2 years later, run it again, and actually control for the multiple hypothesis testing, even among the time points that you run it. And there's a reason for that, is that, you know, if you have a problem early on, and it's an obvious problem, then actually running it early is good, because it will identify it early, even with a small sample size. If it's a large risk estimate, you're going to see it early. But then you're going to miss out on things that are more rare or things that are more subtle. And so we recommend running it early, but then also waiting, and then 2 or 3 years later, running it again. So then what you get after that 2 or 3 years of data is the pickup of all those more rare events, and also those that basically have a less severe elevated risk potentially. Does that answer your question, Charlie?
Yeah, and then just related to that, so you ran each of these drugs separately.
Yes, we ran them all separately.
Could you have run them as a group and aggregate?
Yep.
And then gone down to each one individually? But then, so in your illustration, the ones with the smaller sample sizes didn't show a risk. They didn't show an alert, I should say. Mm-hmm. And if there had been true signals in there, and it didn't show an alert or a signal, would you then move to one of the more standard comparative?
You could from the start choose your, so we ran all those 7 single ingredient statin analyses separately. And we required that a person was incident only to that particular statin. But say you wanted to run them in a group, you could choose to run all of them together and say my exposure of interest is just single ingredient statins and bunch them all. You could probably change your incidence criteria and say you could have to be incident to all statins. You actually could even do that though in the individual runs. You could say I want to see a new user of simvastatin, but I want them to have not had any statin in 183 days prior to their new simvastatin use. So the tool, basically the extraction program, allows you to do either way. And I think that there are different considerations in terms of power that you would want to know before you went in. Actually, for the single ingredient statins, there's quite a bit of power to even see very rare events. And so it's not something to be as concerned with. We did some other analyses that I haven't shared with you today where there's much less power. And there might be a reason to go ahead and go into the groupings. And you can group them as long as you've taken care to how your exclusion criteria work, so you're not double counting people and double counting events.
So I think that'd be important if there's switching, for example, and they move from one product to another.
So we actually haven't done any switching analyses using TreeScan, but there's actually some work methodologically for looking at switching, because that's not necessarily looking at the outcomes, just looking at the behavior patterns that we're doing within Sentinel. So I'd encourage you to go to the website. Actually, so if you wanted to be very efficient about it, I would say go in the upper the search box and look for Josh Gagne. So Gagne. He's the lead author of the methods work related to switching. Okay, we have an online question. The ICD-9 hierarchical tree structure has 4 levels, and the ICD-10 structure only has 2 levels. How is it handled using ICD-10? This is a great question, and one that I didn't talk about today. So we were confronted with the same problem, which is that the ICD-9 tree is actually quite nice, but the ICD-10 tree is really not so hot because there's not that many distinctions. So actually what we did is we used what's known as the GEMS, the General Equivalency Mapping System, and we mapped all of the 910 codes together. And so in our data, we retain whether a person had a 9 code or a 10 code, but we've mapped each 10 code to a 9 code, and when it's required to be incident, it's required to be incident to all related 9 and 10 codes in that time frame. And we did that mapping particularly because we were very dissatisfied with the ICD-10 tree. Another option, though, and one we might consider going forward, is whether to actually just use the ICD-10 structure itself as the tree. So you may be familiar with, those of you who are familiar with ICD-10, it's 7 digits, I believe, and so the first 3 to 4 digits are always required, and then after that you have the option of adding more digits. You can actually just utilize that tree structure itself and use that to run as many levels. The ICD-9 structure is similar in the sense that there are 3-digit ICD-9 codes, 4-digit, and 5-digit ICD-9 codes. We use the multi-level clinical classification system because it groups outcomes that are similar but in different places on the ICD-9 tree. And so we found it had superior performance. You could also use multiple trees, though, as well. TreeScan allows you to do that, so you can stack trees if you would. So some questions. Do you envision any future additions or improvements to ARIA TreeScan? More designs incorporated. We are continuing to increase the number of features, making, so right now we have the propensity score, one-to-one matched analysis, we have the self-controlled analysis, and we are actually making changes to those analyses rather than adding a whole new analysis style to make them more extensible. So for example, we're changing from a fixed follow-up time frame to a variable follow-up time frame, and we're increasing the ability to look multiple times and control for the multiple hypothesis testing, but we're not adding an entirely new design at this time. If folks want to send me their thoughts, though, please feel free. I think our information is available pretty freely. If you look us up on the web, I'm happy to take any suggestions. Some more web questions. Okay, it's going to take me a minute. Yeah, let me read it in my head first. Okay, so this is asking about when we're going to get to an all-by-all analysis is what I think I understand the question to be asking. So right now, as I showed in my slides here, what we've done is I think I called it a 1 to M and an N to 1, but I can't remember which letters I used. Okay, so here, in an exposure-oriented query, we've done one exposure and N outcomes. And we've also done, and we've presented at ICP and other places, many exposures and one outcome. So that's where you take a tree that's built on drugs and you're keeping the classification system of like-related things. So for example, you have a class, and then you have the drugs in class, and then within the drugs in class, you have the dosages and strengths. So you would look at all of those different variations of the drug and look at a particular outcome and see if any of them were more associated than others. So we did this as a test with angioedema. We also did another test with Achilles tendon rupture, and we piloted that analysis. So yes, I think that in the future we'll have dueling trees. We'll have a tree of drugs and we'll have a tree of outcomes, and we will look for all the combinations of the drug and outcome. But there's a lot of considerations methodologically to sort of get through before we get to that point, including redefining the null hypothesis in certain ways. Okay, it looks like there are no other questions from the line. Hi, Claudia.
So I had one more question. So I understand these are acute events, and you mentioned why you wouldn't want to set the risk window ahead of time, which makes sense to me. So then you set an observation period or window, I forget the exact term you used. Do you have any guidance on how long you set the observation window to? Because that seems like that would also be a variable that you should consider.
So currently, in the current, and again, this is a limitation of the current design, I'm just going to go to the overall design diagram to keep it in mind. Sorry, searching for my design diagram. Here we go. That observation window carries with it post-exposure enrollment. And the reason is because the null hypothesis is powered right now to assume everybody has an equal chance of having an outcome on every day of that period. And so that's why we require the fixed follow-up. And in order to do that, because you're requiring enrollment, you're going to end up reducing your sample size. And so essentially the further you push that out, say you want to look for 183 days after exposure or 365 days, you're basically increasing your enrollment requirement as you go, which in the current system will reduce, basically continue to decrease your cohort size. So there's a trade-off there to consider between your sample size and the amount of time you want to look for them. The adjustments that we're making in 2018 is relaxing this criteria. So we've actually are in process of adjusting the null hypothesis itself to allow for people to have any degree, any amount of follow-up time that they contribute. And then that will allow us to then look out longer without paying that penalty in sample size, because now it won't be required. But we have to finish the adjustments to the null hypothesis to make that possible.
Okay, thank you, that helps.
One more from the line. Yeah, so the question here is whether you can take the MLCCS tree and you can remove outcomes from it in what we would call pruning the tree, so that you don't look at things that you think are just going to automatically be noise. So for example, for statins, we could choose to prune out the entire cardiovascular section of the tree and just say anything in the cardiovascular section of the tree I'm going to deem as being the reason that it might show up is because of a prior condition and not necessarily because of the statin. The other thing to keep in mind too is because this is a screening analysis, the idea for it is to make simple choices, so not to think about every single little outcome, and so to actually take a few liberties with pruning out that tree. And so the answer is yes, you can do that, and the considerations for whether you would or wouldn't do that depend on your tolerance for actually seeing those things come up as alerts, versus not wanting to see them come up as alerts and not actually looking for them. So it really is a user-defined sort of specification and something you put in your protocol of whether or not you're looking for them in advance and why. But certainly the MLCCS is prunable. And the and if you want to, as I was saying before, in sort of the materials available to you, this is more related to the TreeScan or the work that I did at ICP. There's a tree there, actually. It's from 2011. It's the MLCCS tree from 2011 that's been pruned slightly to remove things that were not reasonably related to an exposure at all. So things like motorcycle accidents, which are not reasonably related to you having an exposure. Things that were also pruned out were things like normal live births. So it's not the full tree, it is a slightly pruned tree, but you can experiment with the tree, it's available for you. So there's no more questions from the line or the audience. I think we're ready for Michael Wynn to come up and give his closing remarks of the day. Oh, Tyler to do the knowledge check. My apologies. We have to use our phones again.
Hello, everyone. So before we get to the knowledge check, our colleagues out front, Catherine and Margaret, put together some of the metrics from this morning's pre-training knowledge check that I just wanted to share with you, because I know data is very exciting to everyone in the room. So we had about 62% joining us remotely. We had a strong representation from pharmaceutical and biotech industry, followed by government, healthcare, nonprofit, academia, media, and other. Fortunately, everyone was able to remember whether or not they attended the previous training, and about 1 in 5 did. This was interesting. Nearly 60% of folks had never performed a propensity score analysis or a self-controlled risk interval analysis to their knowledge, followed by propensity score matched, both, and then self-controlled or not sure. About a quarter of folks had downloaded or used some of Sentinel's analytical tools. And this was pretty much a bell curve around somewhat well. People generally had some familiarity, but there was room for adding new knowledge. All right. So again, this is the knowledge check. The website, zeetings.com/Sentinel. Access code is Sentinel. And now I go back to the Internet Explorer, right? All right. They've made this Tyler proof, which is very impressive. So, following today's training, how well do you now feel you understand Sentinel's analytic capabilities? Very well, well, somewhat, not well, not at all. All right, so it looks like we got some movement off of the middle into the well section, so that's always a good sign. Would additional trainings on Sentinel's analytic capabilities be helpful? Yes, no, I don't know. All right, that's pretty robust. We can keep going on. With the information from today's training, do you have enough background information to develop parameters for an analysis using Sentinel's propensity score and/or self-controlled risk interval tools? All right, 63% saying both. 20% saying neither. And then a few saying one or the other. Did the live polling feature enhance your experience? Yes or no? Fair enough. All right. 85, that's pretty good. Thank you all very much. Again, I want to thank you for coming, and now I want to invite Michael Wynn, the FDA program lead for Sentinel, to offer some closing remarks.
All right, thank you very much. Wow, that's really loud. I just want to thank all the speakers today, as well as the folks who really made this possible, folks like Margaret and Catherine and Jamila, Anissa, Tyler, and everyone else. I think it's been a great day. I want to emphasize that we really are interested in getting your feedback on those forms. So if you can fill out those forms and just hand them to at the registration desk before you leave, that would be terrific. We've already heard a little bit of feedback already. I know that there were some issues with the parking and directions and things like that, and we will do a much better job next year at putting up better signage. This has all been done in the efforts to open up Sentinel to other users, and this goes alongside some other efforts. So as I mentioned yesterday, there's the IMEDS system where where academia and industry and others can tap into the Sentinel infrastructure and use the exact same tools. We are going to be transforming the market scan data and using that and publishing the ETL code to do that transformation for market scan. We are also going to publish a synthetic Medicare data set online with fully worked examples for a level 1 tool and a level 2 tool. And that is coming I don't know, in a couple months, and that will be on the Sentinel Initiative website. And we will, and as you know already, the tools are downloadable. So we are doing a multi-pronged approach to really, really open up Sentinel and keep this accessible. And the last thing I'll just end with is the slides are coming. They're going to be online by the end of next week, and they'll also be emailed to all individual participants at some point later. All right? So thank you very much.